So, part two of tree biology, the tree biology module is we're going to be looking at some growth and physiology. And again, this is not in any way comprehensive. In fact, this is going to be less comprehensive than tree anatomy and ID stuff. And I'm not trying to, this isn't a physiology course, so we're not trying to learn these things. But I just want to give you an understanding, a little bit of an understanding of how trees grow, so that when we talk about how trees function and some of the benefits we get from trees, we can have some sort of understanding for how those physiological processes actually take place. Um, and so hopefully everyone's been exposed to the process of photosynthesis at some point where plants are able to convert um, carbon dioxide and water and light into oxygen and energy in the form of glucose or sugar. Not going to get too much in depth for this. I think everyone's pretty much familiar with the fact that this takes place, but it's important because trees, they require energy to actually undergo their growth processes, and how they acquire these things is actually important in some of the functions and how that happens. And it happens, at the leaf level where there's chlorophyll. And if we were to actually look at a leaf, this is what the structure is. And you don't need to remember all of this stuff where, there's a upper epidermis, and then the interior is what's called mesophyll and lower epidermis. And, maybe you've seen some of this stuff at some point in high school or other biology courses. Maybe you've taken or plants in general have vascular tissue, in the sense that we have vascular tissue to help us transport things throughout our body. Plants have vascular tissue in the form of xylem for transporting water, and phloem for transporting energy and sugars and starches and things like that. Not going to get too much into that. One thing I do want to point out, though, that I think is going to be important is the presence of a stoma or stomata, I think is the plural there. And this is important for a few reasons. For one, this is where gas exchange occurs for the most part for trees, and so they open up. So there's the stoma or stomata is sort of this hole. It's closed in this diagram, but if it was open, it would be this pore that's on the surface of the leaves, and it's surrounded by these guard cells that open and close the stoma. And so this is where gas exchange occurs. Trees actually open these up so that they can pull CO2 from the atmosphere into their leaves, and then they have CO2 available to them for the photosynthetic process, so they can convert that into energy. The issue here is that, so they have to open up this stoma to pull the CO2 in, but when they do that, they can actually lose water to the environment. So there's usually a higher concentration of water molecules inside the leaf than there is outside in the environment. And some of that depends on it depends a lot on the environmental conditions. So if there's what's called a high vapor pressure deficit, basically that means that there's not much water vapor in the air. So the air is really dry. Then every time they open these up, they're going to be losing a lot more water than if if the vapor pressure deficit was lower. So if there was more water vapor in the atmosphere. And so they have to sort of make this trade-off where, do they want to bring CO2 in, or do they want to conserve their water? And trees usually, kind of like humans, they prioritize their water over their energy. So, similar to humans, you can go, or animals, I guess, in general, you can go, a while without eating much food. You have energy preserves, but you can't go very long without getting water. So water gets the prioritization here, and that's for the primary reason why during times of drought we can see lower amounts of growth in trees is because of this trade-off. They're choosing water over carbon dioxide, basically in the form of how they're regulating the opening and closing of their stomata, and then that has sort of downstream effects for how much growth can take place. And so during drought years, we can see less growth taking place in trees than during wet years. And it depends on what the limiting factor is, but in terms of moisture availability and carbon trade-offs, that's stomata are really important. Okay, so when we're talking about growth, we're not talking about all plants here. We're kind of just taking a look at trees, and so trees grow in a vertical form, and a lot of plants grow in this form where you have this terminal bud. That's going to be the bud, and we kind of introduced this already, where the bud that's at the very tip of the plant form is going to be the terminal bud, and then you have what's called each shoot will have a terminal bud, and then you have axillary buds that occur lower down, and so that's where most of the growth is taking place. And this goes a little bit beyond what we need to know, but there's actually hormones. The terminal bud actually produces hormones that suppresses the growth of the axillary buds, so that the tree actually maintains, for the most part, its vertical form. But you do see differences. Trees growing in a forest are going to have a much more vertical form than trees that grow, out in the open, have a much, less straight up and down. They have more like width to them. A lot of that has to do with hormone production and competition and things like that. So a woody, it's the same, essentially the same processes if you're looking at deciduous versus perennial, evergreen, but where you have a terminal bud, and the difference here is in evergreen, deciduous trees lose their leaves, as we know, lose their leaves every fall and regrow them every year, whereas in evergreen is going to hold on to those needles. No, they don't hold on to them forever. As you move towards the main trunk here, they're actually getting older, and so you'll see needles that have fallen off. Those needles aren't permanent forever, but they last longer than a season usually. And I guess one thing worth mentioning here is they're kind of indicating like these little vertical or crosshair sort of perpendicular lines here. I think are probably supposed to indicate what's called a terminal bud scale scar, where these terminal buds will die off and a new one will form every year, and it'll leave a scar there. So you can actually date, for every twig, you can look for those, and they're pretty noticeable usually. You can see how many years old is that particular twig. When they get larger, when a tree gets larger and you have huge like main branches coming off, it can be harder to see on those once the bark sort of becomes more developed. But on twigs, you can usually see, and you get a sense of how much a tree is growing in a given year, without necessarily having to look at the internal like tree rings. You can just look at the width between each one and get a sense of how much it's growing. Okay, so we've talked about buds a little bit. So if we were to go into that terminal bud and we were sort of to take a cross section of it while it's developing, there are some structures that we would see that they're sort of premature structures, I guess would be a good way to refer to these, in that we can see like the premature structures that are going to form, that are sort of starting to form inside the bud. And so if we take a look here, we have an apical meristem. That's sort of the very tip here, the terminal sort of tip at the bud. It's like the very tip of the bud. And a meristem is, I'm not going to go too much into this, but a meristem is sort of like, one way I think of it, and I don't know if this is exactly accurate, but it's sort of like a stem cell in that other tissues are going to form from a meristem. So meristem gives rise to other sorts of tissues. So this is where pretty much everything is taking place, like cell division is taking place here, and things are starting to differentiate from that as we move away. And so you have your apical meristem, and you have these things called leaf primordia. And a leaf primordia is just an immature leaf. Like these little things in here are leaf primordia, and you also have bud primordia, and it's kind of hard to discern which is which in this diagram. And I'm not super concerned that you are even able to do that. I'm just introducing these concepts to you so that we understand what they are. But leaf primordia, are premature, immature leaves that will form future leaves. Bud primordia are just, immature buds. They will be buds. And you also have the immature vascular system that's forming in here too. And the way this works is that primordia, whether it's bud or leaf, that are closer to the apical meristem are younger. So these ones just developed. These ones developed a little bit a while ago and are sort of starting to develop more so that you can start to differentiate between what is what. So that's what's going on inside of a bud, if you were to sort of look at it. And the entire thing is encapsulated by these bud scales. Okay, so we've mentioned a couple of times the vascular tissue that forms, so I want to kind of talk about this a little bit because it does actually, the distinctions between a woody plant like a tree and an herbaceous plant which doesn't form wood, a lot of it has to do with the vascular tissue. So if we were to take, we have our little plant here, if we were to take a cross section, which if we were to cut it like this, and then we're going to look down the end of the stem, we would see something like this, and we're going to sort of identify what some of this stuff is. So there's one important distinction between herbaceous and woody stems. And so the main one is, well, I guess we'll just walk through herbaceous. So what you have here is like, you have your epidermis in here, and then that's sort of like the outer layer. It's like the skin, basically, of the plant. In between, the very center is what's called the pith, and in between the epidermis and the pith is called the cortex. Now, I'm not super concerned that you memorize what these things are, but what these things are, these little things, are called vascular bundles. And we already introduced these when we talked about, leaf scale scars or leaf scars, and these form bud scale scars. But what these are, bundle scars. But these are vascular bundles, and so this is the primary way that plants transport their water and their nutrients through these vascular bundles. And so you have xylem to the interior, which is used for water transportation, and phloem to the exterior, which is used for sugar and nutrient and mineral transportation. And so that's in an herbaceous stem. They can be arranged differently depending on whether it's a monocote or a dicot, and we're not going to get into all those differences. What I want to point out is that this is not a tree. This does not form wood and does not become a tree. This is an actual tree. So there's some similarities. This would be, it's not labeled, but this would be the pith. You have an epidermis. A lot of this would be cortex. In here, you also have this other ring called phellogen, and I'm not going to really get into what that is. What I want to talk about is this thing called the vascular cambium, which runs between the vascular, or it sort of forms in between the vascular bundles, in between the xylem and the phloem. And this vascular cambium is going to produce what's called, excuse me, is going to form what's called secondary tissue. And that becomes important. So if we were to look, that's, looking at a diagram, this is what this actually looks like. I'm not sure what species this comes from. I forget when I grabbed it. But anyway, this is what it looks like. This would be a vascular bundle. This would be another one. This would be another one right here, and another one right here. But what you can see here is the vascular cambium running along through here, in between the phloem here and the xylem. here. And that vascular cambium is going to produce secondary tissue to the secondary xylem to the interior and phloem to the exterior. And what that is that's actually the wood that's going to be formed. So this is where the wood is formed. And so the difference is that in herbaceous plants that do not form wood, they don't have this. So they just have the vascular bundles. They don't have this vascular cambium, and the vascular cambium is actually a form of meristem that produces the secondary tissue that is wood. And so then that's what wood is just secondary tissue. So technically there's other types of cells in there, like tracheids. They form xylem. So they're formed in fibers, and basically that becomes the xylem is the wood. Phloem is usually is well, not usually, it is what is in between the vascular bundles in the bark and where the wood forms. And actually every single year the phloem gets crushed and sort of has to reform on a pretty regular basis, whereas the xylem exists throughout the lifespan of the tree, and it functions as support. Now, it's not always living. In fact, most of the xylem is not living and does not transport anything. It's just basically there to support the tree. But over time, the xylem builds up, and the newer xylem is obviously functional. So that's how that occurs. Again, I really just wanted to emphasize that the fact that there's that vascular cambium that exists, and that's what produces the wood, and how that process takes place in trees. And it's a little bit similar. We can talk about roots for a little bit. So worth mentioning some things. So you have a root cap that sort of protects the tip of the root and sort of protect it as the root is growing and pushing its way through the soil. You have another meristem that forms the tissue. That's where the tissue forms from. You have like some cell division taking place here, and here you have xylem and phloem in here, and that process is similar to how it happens in the stem. And you have to, if we were to look at a cross section, you have an epidermis, and there's some different things like a pericycle instead of that differentiates from cortex, and we're not going to get into all this. It doesn't really matter. But what's important is, I think worth mentioning, is the lateral roots, which actually don't form from the epidermis. They actually form from this endodermis right here, which is sort of the interior layer. The epidermis is out here. Lateral roots form actually from an interior layer and push their way out from there. A woody root, so that was sort of an herbaceous root, and it's very similar. The distinction is similar here for if we're talking about a woody root. The difference is the presence of, again, a vascular cambium, which is actually going to be producing the wood, and it's going to produce the xylem to the interior and phloem to the exterior, and these little guys that are appearing in here. And so if we were to actually look at what this looks like, this is what it looks like, and you can kind of see that early stage of formation. These are roots, remember, and so eventually, they develop, and you can actually see layers in the wood, which is kind of interesting. Annual layers. So you could date a root similar to the way that you could date the rest of the tree, and a lot of that's due to that wood formation and the presence of that vascular cambium. Okay, so that's really the most of what I wanted to cover. Again, introduce a lot of terms here that I'm not super concerned with you memorizing. I just want you to have an understanding of how that process takes place. There's going to be some reading. So this is the end. There's only two lecture videos for this module. This is the end of those. But there will be some, again, there's always additional stuff after this, so don't forget about that. There'll be a reading which sort of summarizes some more of this stuff. It goes into a little bit more of the development process than what I covered here. So make sure to take a look at that. And then there'll be another discussion question where I'm going to ask you a little bit more in-depth question than what I've been asking in the past. So don't forget about that. So work your way through the rest of the module, and as always, let me know if you have any questions. Otherwise, I will see you next week.